Awesome. Well, thanks for having me. I'm Emily Hicks, president and co-founder of FREDsense Technologies, and today we're going to talk a little bit about our MICA-funded project that we're super excited about. So at FREDsense, what we specialize in is creating portable units for doing water chemistry analysis in the field. So we create small ruggedized units that allow you to measure really specific parameters directly on site, wherever that may be. And so the technology was actually developed out of some work that I did in my undergraduate degree a number of years ago now, as part of a team at the University of Calgary competing in the International Genetically Engineered Machines competition. And this is essentially a worldwide competition run out of MIT that challenges student teams around the world to come up with interesting ways to engineer biology to do something useful or fun, or anything really. And so we were a team from the University of Calgary back in 2012, and that was where the core technology was developed. And so what we developed was this idea of genetically modifying microbes, so E. coli in our case, and turning them into electrochemical sensors for different contaminants. And so the way that works is we go in on a DNA level and we add in a sensory element that allows the bacteria to measure a specific contaminant of interest, and then we allow it in response to produce a certain protein. And that protein's able to interact with a substrate that we add into our test, and it cleaves that substrate and produces an electroactive compound that we're able to measure the formation of that compound at an electrode surface, creating a signal. And this allows us to work in a variety of different matrices, water samples that might not be very clear, might have some color, turbidity, where a lot of optical sensors would fail. And so we took this to the competition back in 2012 and won a huge amount of awards and really naively decided to start a company out of that. And so what we kind of found, when we really looked at this was that we kind of stumbled across this problem that water chemistry is actually really difficult to deal with. And across a lot of industries, not just mining, but from utilities to manufacturing to agriculture, if you want to figure out a really specific chemical parameter in a water sample, you generally have to ship it off to a testing lab. And depending on where you are and what you're trying to measure, this can take many days or weeks for you to get data back, which makes it really difficult to try to make decisions, try to streamline processes, optimize things, decide if something's safe. It's a really big problem. And so we realized that biology might be a really interesting solution to help us solve this. And so we've spent a number of years since then developing the core technology. And the way it works is we create small portable units. We have a cartridge-based system that contains standards and our bacteria that does the sensing. A user puts a water sample into the cartridge, places it into the device, and then that heats up the bacteria to about 37 degrees, does the actual incubation process, and then measures the signal. And you're able to get your results in about 1 to 2 hours, depending on the test. And so this is what some of our early systems looked like. This was one of our arsenic units, and so you can see we have the little box, and then it's kind of a razor blade model where we sell a number of different cartridge kits for the tests that you might be performing. And so the company's first product line was for trace arsenic analysis, and this was an interesting one for us because it was a really well-known biological pathway that microbes detect arsenic because it can be quite toxic to them. And so we took advantage of this and created a unit to do trace arsenic. And we did do a pilot at one point with Ag&Co Eagle, but a lot of this work was really focused on drinking water. And part of that was that we were able to get quite low limits of detection, so that's a real differentiator of the technology. And so we could measure down to 1 part per billion, and arsenic's a really interesting problem because there's a huge number of utilities across North America that have arsenic in their city's drinking water from the groundwater formations around them. They have treatment systems in place, but figuring out if those treatment systems are working, are they overdosing, are they getting breakthrough, they're often relying on off-site labs, and it takes them 3 weeks to figure out what the arsenic levels used to look like in their city's drinking water, which you can imagine is not a great system. And so we launched this arsenic system. This was a case study we did with a utility in Arizona, and we were looking at some different wells across their system's distribution and being able to show that we came quite close to what an analytical lab was measuring for arsenic. But our goal has really always long-term been to develop a whole bunch of different sensors. And so by changing the sensory element that we have in the bacteria, this allows us to be able to develop new sensors using the same hardware software for a bunch of different parameters. And so the company's most recent sensor is for PFAS, so forever chemicals that you might have heard about in the media recently. And so we have a field sensor for PFAS. It goes down to about 70 parts per trillion. And this has been a really kind of timely project for us because regulation just dropped in April in the US for PFAS, regulating it down to part per trillion levels. But what I want to focus a bit more on today is another sensor that we're really excited about, and that's for selenium. And so we worked with a partner called Ginkgo Bioworks, which is the biggest synthetic biology company in the world, and we created a strain of bacteria that's able to respond to selenium. And so it uses all of the same architecture that our arsenic system does, same hardware and software, but just has a slightly different genetic sensory element that we're using. And so selenium is a really interesting, kind of contaminant because it's often present in mining runoff. There is challenges that operators have with having to deal with selenium. And in this industry, there's no really good field technologies similar to arsenic. There aren't technologies that can get down to these low levels that are required for selenium. And so most groups are still using offsite analytical labs, which obviously same problems apply, just takes a long time to get data. And lab analysis for selenium, depending on where you're located, can also be quite pricey. And so there's been some fines recently. Tech is an example of one that was fined up to $60 million for selenium discharge. So it's a pretty significant problem. And so our Fred selenium system, this is just some data that we have. You can see kind of the unit on the right. That's what our newer version looks like. And so this was some data that we collected of some of our selenium system. Right now, we're able to hit about a limit of detection of about 15 parts per billion, but we do expect to be able to lower that. And so this was some testing done of a couple of our different selenium strains. One of the company's major early R&D challenges was being able to store the bacteria for a long period of time because a customer needs to be able to use it. And so that was a big R&D challenge we had. And so this was some data from some trials that we've had on storage of the strain, and we're able to see pretty good storage of the bacteria over a period of time. And we're working on refining this further. And so this shows a little bit of kind of quantitation that we've done. So we're still, fairly early on, but we've been able to show fairly accurate quantitation of some known samples of selenium when we interrogate that on a standard curve. And so each cartridge that we sell has known concentrations of the contaminant of interest in it. And so it's able to plot an unknown on that concentration curve, and that's how we do quantitation. And so for our specific MICA project that we're working on right now, we're hoping to do the last kind of bits of tweaking of the technology and then get it into pilot with potentially 2 different partners. And so we have a couple different groups we're talking about, but that's one of the objectives of the project is to close those pilot deals, hoping to complete some storage, some trials on some storage of the components. So one of our existing challenges is that everything, the bacterial strain we ship, has to be stored cold, which is a little bit of a challenge. And so we have some early data to show that we can actually store it at room temperature. And so we're going to complete some of that work, get it out in the field in 2 different instances, and hopefully gather some critical user feedback that we'll need prior to commercial launch of the system. And so we think this kind of interesting bacterial approach offers a lot of advantages. Because we're using a bacteria, we get around some of the chemical interferences that can be present with other technologies. And so the bacteria have quite a specific response to selenium, which we're able to take advantage of. The technology doesn't have a lot of installation or calibration requirements. It's self-calibrating, and that every test we run has a standard curve, so we can account for the water matrix and any kind of environmental factors that might be present. And you're able to run a large number of samples in a short period of time rather than waiting for that offsite lab analysis. So we're really excited about the technology, getting it out in the field. Really want to thank MICA for this opportunity. It's just really great to be getting funding to support some of this development, and we're really excited to see where things go over the next year and a bit. So thank you so much.